Hi, my name is Dr. Romit Seth. I am a plant biologist and I am here to present some of my research work towards understanding the molecular algorithms to study the crosstalk between tea plant and its environment. Well, before starting, I would like to give you all about my brief introduction. I have recently moved to US and joined North Carolina State University as a postdoctoral research scholar. Well, I am a plant biologist who is aspired to explore an interesting natural algorithms underlying various physiological studies in plants and their interaction with the environment. And prior to joining NC State University, I was working as a senior researcher at CSIR Institute of Himalayan Biosource Technology, which is also named as CSIR-IHBT, which is located in the lap of beautiful Himalayas at Palampur in Himachal Pradesh, India. Let's start this presentation with my research journey. Well, before starting, I would like to thank International Young Scientist Award and ScienceFather groups for providing me opportunity to present in this wonderful platform. During my doctoral research, I was majorly involved in tea research program, as tea is being known for its two major attributes, which is quality and yield, and these two attributes were being affected by three major bottlenecks, which is the self-incompatibility, abiotic stress, and biotic stress. And among these bottlenecks, the abiotic stress, more specifically heat and drought stress, are known to be having a most severe negative effect on the tea quality. And being a part of RKS group, which is run and managed by Dr. Kumar Sharma sir at CSIR-IHBT, I was fortunate enough for being involved in all these type of studies. Well, I hope you all might have heard of tea or chai, which is the most widely consumed beverage next to water. And globally, tea is being majorly cultivated in the tropical and subtropical region of the earth, wherein India is being the second largest producer of tea next to China. India, being the second largest producer, is also among the largest consumer in the world. According to Tea Board of India, the estimated consumption of tea shows a gradual rising demand every year as represented in these stats. Moreover, in India, tea is majorly cultivated in Assam, West Bengal, northeastern region, and Himachal Pradesh. Let me take you to back in history in the mid of 19th century. The year was 1849 when tea was introduced to Himachal Pradesh by William Jameson, who was superintendent of Saharanpur Botanical Garden. The production turned out to be successful in Palampur and Dharamshala region of Himachal Pradesh. In 1882, the Kangra district gazette described Kangra tea as superior to that produced in any other part of India. And by the end of 19th century, Kangra tea has got lot of international attention. Unfortunately, the Kangra earthquake in 1905 had severely affected the various tea growing regions, resulting in abandoned tea farms. And being abandoned for several years, quality of tea was severely affected. And as tea plant is being obligate outcross in nature due to self-incompatibility, it was quite obnoxious for breeders to maintain the quality line of tea. So, here we were interested in understanding the self-incompatibility in tea. Using a tea breeding program, we found two cultivars which were having very high cross compatibility as well as strong self-incompatibility. So, we conducted the field study along with followed by the cytological investigations to identify that these type of self-incompatibility in tea, which seems to be the late-acting gametophytic self-incompatibility, wherein the cross-pollinated ovules seems to get fertilized by 48 hours after pollination, and the self-pollinated pistils they generally withers off. In this case, in tea, and subsequently we performed the transcriptional profiling, and we predicted a comprehensive transcriptional network of this pollen pistil crosstalk in tea. The complete journey starting from pollen germination to fertilization or incompatible interactions were divided into 5 phases in this study, which includes pollen germination, its elongation in the upper styler region, self-incompatible interactions, ovular guidance, and fertilization. Here we identified 182 genes and also predicted a transcriptional interactome network to elucidate the pollen pistil interaction facilitating self-incompatibility or fertilization in tea. Also, we hypothesized that incompatible interactions initiates in the style of pollinated pistils and sustain up to ovary during the late acting gametophytic self-incompatibility. The current study was published in International Journal of Molecular Sciences in 2019. Next study, we all are aware about global warming, right? According to Intergovernmental Panel of Climate Change, which is IPCC, the global temperature has recorded a significant rise from 0.19 degrees Celsius per decade to 0.25 degrees Celsius per decade, which is even faster than the mean annual temperature. And this constantly raising ambient temperature may restrict the spatial distribution of tea cultivation. And hence, we were here interested in studying the heat shock response in tea plant. The current study was published in Horticulture Research in 2021, and I'll be presenting the detailed study in the subsequent slides. In this study, we evaluated 20 quality cultivars by administering the heat shock for different time duration and identified the extreme heat sensitive and tolerant cultivars. Here, we evaluated the heat shock response using the scorching effect. Consequently, we performed the transcriptomics of extreme tolerant and sensitive cultivars. We found 2,831 unique genes exhibiting significant differential expression during heat shock. The gene ontology and enrichment analysis recorded higher enrichment of biological processes in the tolerant cultivar, while molecular functions were higher in case of sensitive one. However, the cellular components were negatively affected in both of these. We also predicted a heat stress associated interlogous network which shows higher enrichment of heat shock proteins and heat shock transcription factors in tolerant cultivar. Here, the inverted red arrows represents heat shock proteins, the HSPs, and the blue one represents heat shock transcription factor, HSFs. A correlation was recorded among HSPs and HSFs with aquaporins, starch metabolism, calcium signaling, phytohormones, CYPs, and ethylene signaling, which probably indicates that there is a well-coordinated transcriptional regulation during heat stress. And then we mapped all our identified key genes, the key candidates, to the 15 chromosomes of T. Subsequently, we performed the pathway reactome analysis to predict the heat stress associated pathway in tea. Here we found that there is an effective activation of HSF1 activation and transactivation and its regulation in mediating heat shock response in case of tolerant cultivar. Also, there is a direct interaction, if you see here in the network, there is a direct interaction of T-specific HSF protein and as well as the T-specific CSHSP90 in the predicted interlogous network, which indicates its key role in inducing heat shock response. Also, we have identified a significant correlations between heat shock associated genes, HSPs and HSFs, with aquaporins, starch metabolism, cellular calcium signaling, phytohormones, CYPs and ethylene signaling, as I have mentioned in the earlier slide. By now, it seems like T-specific CS-HSP90 may have role in heat shock response. And so, first of all, we predicted the structure of T-specific CS-HSP90 protein. And we also found that a chemical named geldenamycin has known to have a specific inhibitory role of HSP90. So, So we performed the molecular docking of our predicted T-specific HSP90, that is CS-HSP1990, using gel-denomycin. And we found here that it seems to block the ATP binding site at the N-terminal domain, which is quite evident in this figure. Performed the leaf disc inhibitor assay using gel-denomycin, the GDA. Here we observed that leaf discs in presence of GDA, when administered to heat shock, they tend to show enhanced heat shock response. This was quite evident with decrease in relative electrolyte leakage and increase in TTC reduction. And also the expression of those genes having direct interaction with T-specific CSHSP90, they were also showed a significant upregulation. So here we hypothesized that in presence of GDA, the T-specific CSHSP90 binds to GDA, subsequently leaving the CSHSF, the heat shock transcription factor, and the activation of these HSFs probably results in inducing the heat shock response in T. So here it seems like there is a well-coordinated transcriptional regulation of aquaporins, starch metabolism, chlorophyll biosynthesis, calcium and ethylene mediated plant signaling system, wherein the heat shock proteins HSPs and heat shock transcription factors HSFs may play a key role in inducing the heat shock response in T. At last, I would like to acknowledge my PhD mentor and guide Dr. Ram sir for his endless support and guidance, without whom all these efforts could not be possible. I would also like to acknowledge my postdoc mentor, Professor Massimo Iorizzo, for providing me the opportunity to be a part of his prestigious group. I would like to thank International Young Scientist Award and ScienceFather group for recognizing me for the Best Researcher Award and considering me to present my work on this wonderful platform. I acknowledge all my colleagues in the lab and from different departments for the help and support which they provided during my research work. Finally, I thank my family and friends for their unconditional love and support. I hope you all enjoyed the presentation. Thanks for being a wonderful audience. Thanks you all.